My name is Scott Lohman. I'm with the Institute for Advanced Learning and Research. I'm the director of the Institute. And it's my pleasure to introduce Dr. Emily Zhou today. And this is another in a series of a set of webinars we've done on plant endophytes. And she'll be focusing on plant endophyte and growth promotion in hydroponic systems. If you're interested in webinars we've had before, please visit our website and our YouTube channel. But with that, I would like to turn it over to Dr. Emily Zhou.
Hello, everybody. My name is Emily Zhou, and I'm a post and doctoral research associate from Virginia Tech, and in the Controlled Environment Agriculture Innovation Center, and here in Danville, Virginia. I got my Master of Science and PhD from Virginia Tech and graduated in 2020. And I have been working in the hydroponics, leafy greens and vegetables for over 2 years. Here comes my presentation. And hello, everybody, and welcome to my webinar. And today my talk will be exploring the potential of beneficial bacteria endophytes for hydroponic lettuce growth promotion. And I am Emily Zhou, and I'm a postdoc research associate from Virginia Tech. And the Plant Endophyte Research Center, and here in the Institute for Advanced Learning and Research, and has done a lot of work for the past 10 years. And I'm glad to be one of the team for the past year and to do the field research on hydroponic growth lettuce. My outline today, and I will first talk general introduction of bacterial endophytes, and then I will talk about our new Controlled Environment Agriculture Innovation Center, and here located in the campus of IALR in Danville, Virginia, and what we have done and ongoing research. And the last, and I will talk about the research we have done and ongoing on growth promotion of hydroponic lettuce, utilizing the beneficial bacteria endophytes, and end up with question and answers. And what's the definition of endophyte? And an endophyte is a naturally existing microorganism and is naturally occurring. And it is an endosymbiont, often could be a bacteria or a fungus that lives within a plant body. Also, and it's living with the plant but not causing any disease of the plant. It benefits the plant host and by promoting plant growth. It also increasing the nutrient acquisition and increase the tolerance of stress, and also increasing the pathogen resistance. And here is a strategy of the endophytes library. And here, and we have done by the plant endophyte and research team. And so far for the past 10 more years, and they have done with more than 2,000 bacteria strains and discovered a lot of useful bacteria endophytes can be used and in the apply field. And why the bacteria endophytes can promote the plant growth? And because they can increase the plant hormone auxin, and also they can solubilize the phosphate and which phosphorus and is a plant essential and fertilizer, and so for plant to uptake. And also they have nitrogen fixation, and also they can active the ACC deaminase. And ACC deaminase is a bacterial enzyme and promotes plant growth by lowering plant acetylene levels. And also, they have a sulfur. Sulfur is a small, high affinity iron chelate compounds, and which are secreted by microorganisms such as bacteria and fungi for plant uptake or for the plant pathogens. And this in this ender, and you can see this table, and they are the four plant the bacterial endophytes we are using later on, and their here's their traits you can see, and then they have all of them, they have a phosphate solubilization, and they all can produce and oxygen, and some of them have a nitrogen fixation function, and some of them have high ACC deaminase, and all of them had sulfur. And the picture here shows the plant growth promotion in vitro and in the lab testing with Kentucky 31 tall fascule grass. And you can see the PAKM is a well and known now bacterial endophytes, and we were using. It can increase the growth of the Kentucky 31 tall fascule plants a lot by using that compared with not using that. And the plant endophyte and the research team also have done some lab, greenhouse, and field studies. They're here showing some field study on the strawberry plants, both in the greenhouse and in the field. And Dr. May has published a paper about this if you are interested in just recently and in 2021. And here is our new established Control Environment Agriculture Innovation Center. And it is located in the IALR campus in Danville, Virginia. And it is a partnership between the School of Plant and Environmental Science and at Virginia Tech and Applied Research Division of the IALR. And we have conducted several researches here. And first, introducing the Dutch buckets and gutter systems. And this is the first research we have done here in the center, in the CEA Innovation Center. We grow strawberries both in the gutter system and Dutch buckets. And also the Dutch buckets can grow the CBD used hemp plants. And here is a greenhouse, and we have 20 units, systems of nutrient field and technique systems we call NFT. So this make us possible to do several treatments and several replicates of the different treatments. And we have growing a lot of lettuce plants, and also it can grow other leafy greens like basil. And here is our indoor vertical NFT systems. And I have involved in the building up with the indoor vertical systems. We use artificial LED lights. The good things about this is, and it can run 365 days and in a year, not limiting with the weather, lights, or other conditions. So the indoor area, we can keep a constant temperature, and which normally, and we set up at the optimal temperature for the plant to grow. We have 6 and NFT vertical systems which can grow the leafy greens, and also we can grow micro tomatoes. We have successful grow micro tomatoes and to the ripe stage. And we also have 2 open gutter microgreen systems, and we were using a burlap as the growth media, and it can grow microgreens and also can grow baby leaf lettuce. And many more I haven't shown. And now coming to the third topic, and our the endophytes studies. Coming to the endophyte studies on hydroponic lettuce. And the first, we had a big study about the screening of different endophytes on hydroponic lettuce plant growth promotion, both in the greenhouse and in the indoor vertical systems. And for both the greenhouse and the indoor vertical systems, and we use 4 different types of endophytes. And previously we have introduced and the 4 endophytes, their traits and why they can promote plant growth. That's PAKM and IALR number 632. IALR number endophyte number 1089, and the IALR 1379, and plus the control. We applied 1 ml of endophytes and at the true leaf seedling stage, and normally that's after a week of the sowing the seeds. And we use 6 lettuce cultivars and Nancy and Butterhead racks. And as the picture showing, you can see they are Butterhead types and they can form a head. And Salanova red oak leaf and green oak leaf and red sweet crisp, and these 3 are the loose leaf type, and red rosy is the romaine type. And we have 4 blocks, and in the greenhouse that depends on the different light levels. And also we have 3 blocks, and which is 3 individual vertical racks. And each block has 6 plants, the replications. And we use our Virginia Tech and lettuce fertilizer, and at EC at 1.7 and pH keep as at 5.9. And for the greenhouse and NFT systems, and we grow the lettuce for 22 days after transplant. And last year in the springtime, and the greenhouse has natural light. We didn't put any supplemental lights. Our watchdog data showing the mean light level hours per day, and it's around and 8 to 9 hours. The mean daily light interval is 25.5 mole per meter square per day. And the temperature and the mean temperature is around 68 degrees Fahrenheit. And with the mean day a little bit higher and night and to 65 higher to 72 to 65. And the relative humidity mean at 63%. And we use ambient CO2 level. For the indoor vertical NFT systems, and we use LED grow lights, and they are constantly and with the PPFD at 250 mole per meter per meter square per second, and we keep 18 hours light per day per 24-hour cycle, and so we have a daily light interval of 16.2. And temperature constant at 65 degrees Fahrenheit. And we have a mean and relative humidity of 56%. So we have 2 dehumidifiers to keep reducing the humidity. So when our plants are running, the humidity could be very high. And we also have a CO2 tank pumped to the area. So make sure the CO2 level is above 400 parts per million, and which is equals to the atmosphere. Higher than that. And here is the result. And for the greenhouse Newton field technique systems, and we find out most of the 6 lettuce cultivars and all have growth promotion and by some or all of the bacteria endophytes. You can see this more obviously by this bar chart, and the one with astral, that's the significant increase. So this, the left one, this, the gray one is a control, and all other 4 with endophyte treatments. So you can see most of the some or all of the endophyte treatment can benefit the lettuce growth of 6 cultivars. And here shows the growing percentage, and like the Salanova green oak leaf, and using the endophyte PAKM and number 632 can increase more than 50% of the fresh weight. So that's a big benefit. And somehow the indoor vertical systems are more complicated. And we see, we only show the butterhead racks and showing the significant growth. And our other cultivars, they need more research. And I think there's a lot of factors, environmental factors, and also with the environmental factors, with different light level, or we did not have enough replicates and to show the significant data. So And from the big screening, and we have addition studies, and we pick up 3 cultivars, lettuce cultivars, and also we pick up the most advantage plant bacterial endophytes and for this additional studies. And this study was conducted in September to October last year. And we reduced the EC to 1.5. And because, especially in the indoor and vertical systems, we found some of the tip burning of the plants. So, and we try to get the higher quality of the plants and still keep the pH at 5.9. And this time we grow them for 4 weeks, 28 days. And we do harvest every 4 days and after transplant. For the greenhouse, and we use 3 endophytes, the PAKM 632 and 1089, the most significantly increasing from our previous study, and the 3 cultivars which is significant showing the plant growth promotion is the Rex, Salanova green oak leaf, and the red rosy, which is a romaine type. For the indoor verticals, and we are only using 1 endophyte, which is 632 plus the control. And we choose 3 cultivars, butternut rex and green oak leaf same. And we add another cultivar, Salanova green butter, because we had other studies showing the significant growth and in the indoor vertical system of that. Here is the result, and we only see 1 cultivar, which is Salanova green oak leaf, shows significant growth, and after from 20 to 24 days, and the DAT is abbreviation of days after transplant. So, and especially the Endophyte 632 and showing the significant growth promotion at the late stage of the plants. And normally, we harvest during this time. And the indoor vertical and NFT system is also showing the cultivar Salanova green oak leaf has significant plant growth after 8 days and after transplant. And so they have plant growth at 24 days and also 28 days. But because of the sample we are using, and we only each time, and we do not have a big system can grow more plants. So every time we only harvest in the indoor vertical system, only harvest 12 plants. And I believe, and if we have more plant samples, more replicates, and it will show the significance. And the conclusion of our bacterial endophyte study on hydroponic lettuce, and the beneficial bacterial endophytes, they can promote the upper ground shoots growth of a greenhouse hydroponic lettuce plants. And just we need more research and in the future, and especially on the indoor vertical systems. And here on the right, the picture is our ongoing endophytes study in the greenhouse. And we are using 1 cultivar, the significant cultivar previously I shown the Salanova green oak leaf, and we are using the endophytes and IALR 632. So and today is it's 1 week after transplant, and we will see this time we have more replicates of plants, and we're expecting significant results. And that's all. Thank you for listening to me, and I'm ready for questions.
Okay, thanks so much, Dr. Zhou. We're really excited about this research in hydroponics. In the past, most of our research with endophytes has been done either in the lab, greenhouse, or the field. And we always saw terrific results in the lab in our in vitro screens. And then, of course, they were reduced a bit when we took them to the greenhouse and put them in potting soil. And then even further, the beneficial results were reduced in the field. But we feel that the hydroponic systems in the greenhouses are much more like in vitro systems. So we were, again, very excited about moving into this area. And we'd also like to thank Dr. Michael Evans for coming down and partnering with us to create the Control Environment Agriculture Innovation Center where Dr. Zhou works. And with that, I'll sort of open up the floor for questions. We do have one that's been submitted already, but feel free to enter the chat to share your question with us. The first question we have is, what considerations should be taken for lettuce cultivation in hot, humid climatic conditions, sort of like tropical climates? And this, Dr. Zhou is much better to answer this than me.
And very good question. And I think lettuce is a cool season crop. They don't like hot and humid environment. But our greenhouse condition resembles the hot tropical weather climates because it's always hot and humid. And what we do is, and we try to, we have cooling pads in the greenhouse, and we also have a ventilation, and we have a vents, double, triple, fourth, the vents. So it's set up when it's above certain temperature, and it will start running. Also, and our greenhouse is pest free always when it's hot and humid. You increasing the possibility to get pathogen. And our greenhouse, both in the greenhouse and the indoor vertical area, are pest free. What do I say that? Pesticide free. So we do not use any pesticide, and it is strictly, and so we have very, very strictly sanitation process. Everybody enter the way it must be sanitized. And so if we see any indication of a pathogen, and we discuss all plants. So, and we don't use any chemicals or other ways. So far, so good for the lettuce. I think, and I don't know if anybody doing hydroponics. I heard in the tropical area they do the hydroponics outside, but most hydroponics I have seen are in the greenhouse condition. So I hope this answers the question.
Okay, thanks so much. We have another question, which is a terrific question. The questions we like to hear is how can I arrange a visit to see the CEA Center? The Institute is open to the public from 9 to 5 during the week. Please feel free to reach out to me at scott.lohman@ialr.org. My name can be found also on the website as well as Dr. Zhou's. Please feel free to reach out. We have wonderful facilities. We are proud to show them off. And I'm open anytime for a tour.
I also, I didn't mention in the presentation the good things of growing hydroponic leafy green, especially the lettuce, and is the hydroponic growth lettuce has a very, very long shelf life. So, and I brought one today. It's Sananova and red butter. And I harvested this one before Christmas. So it's been 6 weeks. And so they are still crispy and nice and to eat. I also not only lettuce, also for other like spinach and very normally if you buy the grocery stores, they are life maximum like 1 week, and it can also stay in the fridge for more than a month.
Yeah. And we're very excited about moving into the CEA area, the Controlled Environment Agriculture area. Again, it's a huge opportunity for us. It fits well with the Endophyte library with biologicals. We're not only looking at growth promotion, but we're also looking at disease control. We have another scientist here that focuses primarily on disease control. He develops pathoassays. So we're looking at various typical problems with indoor crops and different ways to control pathogens biologically. But just in general, we're excited about the controlled environment ag movement and how it's growing in the region and beyond, not only because of the freshness of the vegetables, but because it's also regional, it's economical, it can be produced year round. So it also provides opportunities for the farmers in a region. So in a number of different areas, we're excited to be in controlled environment ag and applying our endophytes as a toolbox. So with that, I don't think there are any more questions, and it's after 2:30, so we'll wrap up the webinar. Thank you so much for attending, and please visit our website.